Well, let's talk a little bit about the carbon dynamics in Florida rangeland and how management and environmental fluctuation affect it. So I'm presenting today, but this is a work, it's a huge team from UF in the Arch Bowl that help on putting all this information together. So rangelands in Florida extend for more than 3 megahectares, and these are associated to longleaf and slash pine with grasses and saw palmetto and small trees. And around 1.6 megahectares of non-forested areas extend from the center of the state all the way down to the Everglades. And what is important on this is that this area hold 2/3 of the state's cattle, having a huge impact on the food industry and the economy. These rangelands are managed by combining prescribed fire and mechanical brush control, basically to improve the forest quality for cattle. These rangelands, they have a lot of benefits. Many benefits. They support livestock, water conservation, wildlife, carbon sequestration, and many others. So they're really, really important from many point of view. These rangeland are used in the beef industry, as mentioned before. They have a high biomass production and a natural fire frequency between 3 to 4 years. And managers, they use the prescribed fire in about every 4 years to improve, like I mentioned before, food quality, forest production, control wildfire, and reduce the presence of saw palmetto. Based on this brief introduction, these questions, many questions were asked about how management of the Florida rangeland, including fire and mechanical control, combined with the environmental fluctuations, affect many things, basically the functioning of these ecosystems, including carbon storage and carbon exchange dynamics, and that includes the biomass production, the net ecosystem production of NEP, the gross primary production of GPP, and ecosystem respiration, including the soil respiration, seen here as ecosystem R-eco and R-soil. Also, how it affects evapotranspiration, species composition, soil and carbon nutrients. I do not intend to go through all of these, but this is just, a mentioning of the many questions that are being that we're looking to respond with this ecosystem. So to go through that, there is a large scale experiment down at the Cattle Research and Education Center at ONA. The experimental area is a randomized block designed for blocks for experimental units or treatments. And you can see here the 4 units with different colors. And we have an onboard or control plot repetitions inside the different blocks. The business as usual, which is a 4-year fire cycle treatment. This ASP aspirational 2-year fire cycle, and a 4-year cycle combined with chopping. There is an eddy covariance tower down here in one of the business as usual units, and you can see transects inside each unit. Those transects are used, 50-meter transects, and are used for sampling biomass and soil sampling for further analysis and greenhouse gases emissions measured at the soil level. Now, what do we expect in terms of carbon dynamics? I'm going to be focusing basically on the carbon dynamics today. So based on studies in other ecosystems in the region, we expect that these South Florida rangelands are carbon sink under current management practices. And those can be a carbon source, at least during the 5 years in extreme drought years. We expect that vegetation recover quite fast and based on the carbon uptake after the fire. And we also expect an increase in greenhouse gases emissions and say CO2 from from the ecosystem in general after the fire. So we base that on this slide that shows a nice transect of the carbon dynamics across the landscape in Central Florida. And I thank Dr. Ross Hinkle for providing this slide to me, which shows this urban ecosystem near to the UCF campus being a carbon source. So it's 11.6 basically ton of carbon per hectare per year, and we go from the Bahia grass at Judo Ranch with 1.26 ton of carbon per hectare per year in a 3 years study, and scrub oak with about 13 total years of study, and so on. And we have this ecosystem here, this large pine, the Kennedy Space Center and Disney Wilderness Preserve that act as a carbon sink between like 1.3, 1.5 to 4.2. So we expected that this ecosystem, this rangeland at Ona behaves as a carbon sink between whatever the scrub oak is doing and this large pine. So we have this all the way down to sawgrass and Spartina marsh and salt marsh at the Kennedy Space Center. We don't have Ona rangeland here, but we will have it soon. So we expect the ecosystem is a carbon sink in this 4 years treatment, carbon sink in that time. I'm going to be talking about the net ecosystem carbon exchange, or NEE. So that is the carbon flux measured as CO2. We will talk about ecosystem respiration of RECO, evapotranspiration. I will just mention that evapotranspiration, sensible heat fluxes, using the eddy covariance approach. This system down at Ona was established early 2016, so we are approaching to 5 years of it. On a business as usual unit, the 4 year fire cycle, we had a fire in May 1st of 2019, and measurements are running until today. So if we have the chance, we can connect and show the current variables that are happening there in the last 30 minutes, carbon flux and ET. So a little bit how eddy covariance work. So we have wind blowing on top of the vegetation, and whenever it blows over the vegetation, it creates turbulence. So those are the conditions for eddy covariance, and that turbulence creates many eddies, which are these kind of circles that we have here. Those blows through the eddy covariance sensors, and each one of those eddies carry a CO2, an amount of CO2, an amount of water. It has a temperature, it has a velocity in 3 directions. And with that information, we collect that, and we proceed to estimate fluxes of any entity that we have at the sensor, the appropriate sensor to measure it. So it can be CO2, water vapor, methane, and there are some nitrogen sensors that can be set for eddy covariance as well. So the ONA setup, the core of the eddy covariance system is an Irgason from Campbell. It has the open path system that measures CO2, water vapor, which is this sensor, this one over here. And this sensor here is the sonic anemometer. It collects data for CO2, water vapor, wind directions, wind velocity in 3 directions, X, Y, and Z, and air temperature at 10 Hertz, quite fast. We also have an array of meteorological sensors to measure air temperature, relative humidity, this sensor over here, radiometers, long and short waves in both directions coming in and going out, and an array of soil sensors. We have a phenocam here, and we have a lot a power and acquisitions in data storage system. So we collect that at 10 Hertz, and it's collected 24/7, monthly, yearly, and we have 4.5 years of data that I'm going to show you in a minute. So once that data is collected, we bring it to the lab, and do a post-processing. We do quality control in the high frequency data. I'm not gonna go deep in those details. And with that, we estimate 30-minute fluxes of CO2 as NEE, latent heat, which is evapotranspiration, and sensible heat. Once that is estimated, we apply several corrections on that data, and we use a software called EdiPro from Reco to do that. Please keep in mind the convention that EdiFluxers use is that negative carbon fluxes indicate uptake by the ecosystem, and we apply that for NEP and or NEE, and excuse me, and gross primary productivity as well. So once that is processed, we go ahead and go to gap filling meteorological data. We check for the energy balance, and we proceed to do the NEE gap filling. We can, we filter data based on rain, equipment malfunctioning, eddy covariance conditions are not good, so that means there is not enough turbulence for an exchange. And after that is done, we go to partition. And remember, what we estimate all the time is NEE. During the day, NEE is the balance between GPP and ecosystem respiration. And during the night time, NEE is basically ecosystem respiration, at least that we work on a pineapple plantation that is a camp. Once we do that, we proceed to do partitioning into GPP and ecosystem respiration. And for that, for this ONA data set, we are using Toby, a software from Reco. When we have the 30 minutes gap fill and measure NEE, we integrate those 30 minutes into daily, monthly, and yearly NEP. The other parameter that I want to mention to you here is the GS max, which is the maximum canopy conductance. And I mention this because it's one of the biological factors that we use later to explain variability in fluxes that I will show you later. And we use that by inverting the Pigman-Monteith equation, since we have evapotranspiration already measured. A nice thing about ONA, and this is great, ONA joined Ameriflux early in 2020. It was by February, March, if I remember correctly. So, and the data is already published and available for the last 4 years of data. And I'm gonna try to connect here pretty quick to show you a few things about this ONA site. Just please bear with me for a second. So this is the overview of whatever is happening in there, all the information. Something important here, you can see we have so far a DOI for the ONA data set. So if any person uses it and does not, and Maria decided, we decide not to be co-author in any review that is using this data, so they have to use this citation and use the DOI. This data use log, if you can see here, this data became available early September, and by now, Maria should have gotten over 50-something emails telling her that this data is being downloaded and being used. There are pictures that you can go in there and check and download the pictures. There is something really nice about this NDVI that Ameriflux put together, and you can go in there and look at from the beginning of the experiment, of the very beginning of the MODIS data collection. Publications, we don't have a publication there yet, but it's coming soon. And we have this bottom. Bottom is the biological ancillary disturbance and metadata for this site. So that can be checked here and downloaded. And something else that I want to show here is this data processing status. So it's going to show my all sites that I'm related to, and I just want to show honest data set. For this, you see this green, so that means data is available, and it became available not that far ago. So that is really, really nice, and you can check more on the Ameriflux by going into ameriflux.gov and look at all the benefits of being part of Ameriflux. And I've been benefited a lot with quite a few synthesis papers from the sites that I'm associated to. And Ameriflux provide calibration guides for free, visit to the site for free, technical advice, and more than that. So the other thing that is being measured down there is the soil greenhouse gases emissions at 14 days interval from 8 a.m. to 11 a.m., which accounts for the diurnal greenhouse gas emissions variation. And there is a complete protocol to collect and analyze that using a gas chromatograph system. So on this slide, I'm showing you what we are using to gap fill data that is being collected every other week over there. So we are using this random forest strategy. This is an algorithm that creates data sets, bootstrap data set, and generates independent regression trees using variables at each split node. And after that, the random forest aggregates the prediction from individual trees. So, and the two variables that are explaining over 83% of the variability of the carbon, the CO2 fluxes for the three units at business as usual are the soil temperature and the soil water content. And again, this is the model performance. And the gap fill data for the 2 years that we have pre-fire and post-fire, it will come in the table that I will show you soon. So we are working, applying the same strategy for methane and for N2O fluxes. So that will come soon and will be part of the manuscript that we are working on right now and should be submitted soon. So let's show some results. Let's start with climatic variability. So we can see a tremendous climatic variability during these 4.5 years of study. So if we look from the end of 2016 into May, June of 2017, which is this part here. So the bars are the monthly precipitation data, and these lines are the long-term average for each month. And we can see that the system went under extreme drought. The perma drought index went below -4, and extreme drought, which can be seen here in the topsoil water content. And during that extreme drought, the topsoil, the first 20 centimeters, went under or below the wilting point for that year. And something similar here, but less severe. And the year that we had the fire, look at the ecosystem had average to above average precipitation. The other thing that I want to show in this graph is the water table depth since the sensor was installed. And look at this. Once the water table goes below 1 meter, the ecosystem, the topsoil in the ecosystem gets to below the wilting point, which is a really drought condition. So some results, again, soil and air temperature, soil temperature higher than air temperature. And of course, it had an increase. The fire happened here, and it increased well above the air temperature in the following month. So this thing here is the 30 minutes, 4.5 years, from January 2016 to June 30th on 2020. Each dot, it's 30 minutes NEE measuring gap fill. So it's hard to see anything in there, but we can see fluctuations, and here is the fire signal for that. So I don't want to stop that much in there. When we have these and we integrate all of these data, like I mentioned before, daily, weekly, monthly, yearly, we come up with these annual carbon fluxes, grams of carbon per meter square. So we have 2016, 17, and 18. Look at how 2017 was kind of a little below the previous year, which was really wet. And we have an average of pre-fire condition of 368 grams of carbon per meter square. Remember, negative means carbon uptake. And the fire year, the ecosystem had 182 grams of carbon per meter square during the fire year. Remember, the fire happened May 1st. GPP, however, during the fire year increased about 200 grams per meter square on the fire year, and ecosystem respiration increased about 400. So soil respiration, and this is the data that I'm going to show, so the only data that I'm going to show in this presentation, it increased by 200 grams per meter square during the fire year, and we have ET and precipitation. Just to mention that we have that data, it's already processed, but I don't have the time to go through all the ET, which can be another presentation like this one, or the water use efficiency that can be in part of another presentation. So we had that yearly carbon fluxes, but we want to look at the fire effects on carbon stocks. And remember, the units are grams of carbon per meter square. So before fire, the vegetation, what the folks down there did, they collected that information on those using those transects that I've showed before. And the vegetation had 213, 214 grams of carbon per meter square and 20 grams down in the litter, and there's a huge amount in the soil carbon. So fire happened, and we are taking the approach from Alexis et al. Say similar ecosystem nearby inside the Kennedy Space Center, and we're assuming that 79% of that vegetation was affected, which is 168.7 grams. However, not all of that carbon was released to the atmosphere. And the carbon litter, we are assuming 69% of that got affected, which is 13.8 grams. So next day after fire, this is great. They went again out there and collect litter and collect the amount of material that went down into this litter. So if 13.8 grams, so these 50 that is over here in liters plus ashes, that was measured. So there was an increase in litter from 20 to 50. So if 13.8 grams were released to the atmosphere, 6.2 came here. So, and the difference of that is this 43.8 came from the vegetation that was not released to the atmosphere and came like a semi-burned material and ashes down to the litter compartment. Whatever was left in the vegetation was this amount of carbon, and the system, if we add these two, it's 94, 95 grams of carbon per meter square. Of course, this can be modified. You can give me arguments against this or in favor, and but it few things matches in there. So this is estimated as a difference, but this was measured and the numbers are really, really close. The difference is not more than 3 or 4 grams per meter square. But what is really, really important in terms of management is this. 4 months after fire, folks down there went and remeasured vegetation recovery. And look, 4 months after, 200 grams per meter square were already there. So 4 months after, vegetation was back to the initial values before fire. Now, having these, whatever was released by fire and the tower, we combined those two and we tried to estimate the carbon balance in a 4-year fire cycle unit or business as usual treatment. Again, the units are grams of carbon per meter square. If we do have the average, the table that I showed before, pre-fire average NEP minus the 5-year NEP, we come with the loss in productivity. So it was 181, but it should be this amount. So this is the loss in productivity due to fire. But if we integrate the 4 years NEP, so we have this 12.8, 12.9 ton or 1,287 grams of carbon per meter square. If we subtract the loss in productivity, we have this number here. And if we subtract the loss by ignition, we come up with this number here. I'm calling it net ecosystem carbon balance. It can be also called net biome productivity. There are many arguments of using one or the other, but it's the carbon balance after the 4 years and how the fire affected that ecosystem. And we come up with this huge number, which is really nice. So it's 961.41 grams of carbon per meter squared. So if we want to convert that into biomass, that is roughly, let me tell you that in terms of tons of biomass per hectare, is about 19.2, 19.5 tons of biomass per hectare in those 4 years. So, but we look so far on the yearly, on the 4 years, the yearly carbon fluxes, and it's not possible to explain interannual variability with 3 years of measurements, and sometimes even with the decade, with 10 years, 15 years, and I've even seen more than that, and it's difficult to explain interannual variability because when you try to interact with management, fire, and climatic fluctuation, the number of years that you end up on each one of those is very low, like 3 or 4 in each one of those. And we try to look at the higher scale, like going to seasonal, monthly, and daily carbon fluxes. And I try to go through seasonal in this case. This is what I'm showing here. I like this graph a lot because I separated the cumulative carbon fluxes starting on January 1st all the way to December 31st in 3 different periods. So this first one here is all the way from January 1st to the end of April. This one is 4 months, this one the following 3 months, and this one the following 5 months. So look at this. The variability here doesn't matter how climatic fluctuation is having or is happening there. The effect seems to be that carry over the following 3 months when the fluxes separate. Look at 2017. So it was accumulating carbon, and all of a sudden it became carbon source, and after that, keep accumulating carbon. And look at 2020. So the first 6 months of 2020, like nothing happened to this ecosystem the previous year. And fire happened here, so there was a carbon release, but it took not that long for the ecosystem to become like a carbon sink or start incorporating carbon again. So I look at the average daily carbon fluxes for these 3 periods. Look at the day 1 to 120, that is to the end of April. Average NEP is 127, so it's the highest relatively compared with this, probably not different, but when you accumulate them, you come up with different numbers. What I want to emphasize here is that in this period, this one here, the ratio R equal GPP is like 0.72, and here is 0.85 for these other 2 periods. This part here, it's 40.6 grams of carbon per meter square per month in average, which is 44% of the annual NEP. This came down to 24. Of course, this is 3 months, and it's close to 20% of the annual NEP, and these last 5 months here is only 36% of the annual NEP. So I like this graph a lot. If you go from the seasonal to monthly, this is the monthly NEP. Remember, negative means carbon uptake. Few things to look at these. The months with the highest carbon uptake are March and April, and we have August and September here. June of 2017 here was a carbon source. And remember, the previous graph, it did not have an effect. It was not affected in the first 4 or 5 months, but June was affected. Fire happened here May 1st, and the ecosystem changed from being a strong carbon sink about 35 to a strong carbon source about 60. So there was a change, a shift in more than 90 grams per meter square in that month. But look, by June, it was half of that. And by July, look, it was even higher than July 2017, which experienced a strong drought. And by July, the drought condition was over, but there was kind of a carryover or lag effect from the drought. And look at 2020, I put that on blue, and it's like nothing happened. And even here, and what else here, it's higher than the other, February from the other months. So what now GPP, so after the fire, even that it was high here in April, but after the fire, GPP was reduced by 50%. And after recovery, we had the highest, by a lot, highest GPP values in the ecosystem. And our eco, something interesting to look at this is it stayed kind of stable during the first 4 months and after increase. And after the fire, even that April and the fire year was higher, but it increased and stayed high for several months of the rest of the year. So GPP was reduced, but ecosystem respiration increased. So drivers on that monthly carbon fluxes, I use a multilinear approach here, and of course checking for collinearity, and for NEP, global radiation and precipitation explain only 30% of the monthly variability in NEP. Of course, I excluded the fire months and included like November, December of the fire year. But for GPP and ecosystem respiration, soil temperature and water content explain about 63% of the variability, which is high. If we go into the daily carbon fluxes, the top one is net ecosystem productivity. So these gray lines, these gray bars are the daily values, and the dark one, the black one, is the 7 days average. And we can see here this carbon source over here, this carbon source over here, and here we can see the fire effect. So the system became a carbon source, but quite fast it started recovering, and 71 days later we had a week, complete week during, and the ecosystem was a carbon sink during that week. So that's the time that it takes to recover. This one here is GPP. I inverted the scale. So the daily GPP, and the green one is the green index from the phenocam. Let's see how GPP tracks that. And here is the fire signal in this, but after the recovery, GPP is the highest, and the green index as well. And here we have the ecosystem respiration and soil respiration tracking it very nicely. Now, what's happening there? How is the ecosystem photosynthetic capacity being affected and recovered because of fire? I use these very well-known live responses and fit NEE against the photosynthetic active radiation, or PPFD. And here is a parameter that is called GPP optimum that I'm going to talk about it, and it's the not the NE, it's the GPP at light saturation. And that week before the fire, it had this number, negative 25.8. Remember, negative is carbon uptake. But that week in May, it was to this point. But June, it was already between May and the last week of April. By July, like nothing happened. Of course, the quantum GIL changed a little bit here, and the dark respiration as well, but I want you to focus on this. Look at that. July, basically, it was back to normal situation again. And by September, we had the highest optimum GPP measure during the 4.5 years of this study. Now, we went through yearly fire affecting carbon stock and fluxes, seasonal, monthly. Now we are here into the drivers for daily carbon fluxes. We use the tree regression. The tree regression is a non-parametric class of regression models that performs splits on the dependent variables based on the different variables in there. It does not overfit the models, and what I want you to focus here is that for daily NEP, and we intentionally left here this bond data after the fire, and the same over here and over here. So this tree regression approach goes splitting data, and the main split, the main variable is the global radiation. daily global radiation. This is in megajoules per meter square per day. And the same for GPP, it made the first split around 15, and for ecosystem respiration, as we expect, is soil temperature. But please look at the huge variability after that first split, and that is where we go to this. It looks messy, daunting, but if you follow me, I will walk you through it, and it's not that bad. So, like I said before, the first split for daily NEP happened at global radiation at 9.7, 9.8 megajoules per meter square per day. So this first split, below that, the ecosystem was always a carbon source. Now, look at the number of days on each node. So we have plenty of data after 4 years, 4.5 years. Of course, I'm not including here the 5 months. I exclude the 5 months, like from May all the way to October. The second split, it's again global radiation between 9.7 and 19.8. So here we have a combination of soil temperature, global radiation, VPD, which was not in the monthly factors explaining it, and we have soil water content coming up to explain the NEP, the daily NEP variability. In this box, we have 25 months of data. If you look at each node, 25 months of data on the daily basis explained by these variables. And if we come to the next, whenever global radiation is greater than 19.8 megajoules per meter square per day, global radiation, soil temperature, and look, we have biological factors showing up here. This is the stomata opening at the canopy level, and we have green index, which is an indication of the biomass present in there. And we have combination of those explaining this part of the daily variability in NEP. And this part here represents more than 18 months of data that we collect. So for GPP, remember the first split, it's at 15, and we have that in there. Look at how GPP goes increasing all the time. And we have this first split here, and global radiation combined with soil water content, VPD, soil temperature explain the increase in GPP over here. The next one, we have the green index showing up, soil water content. And for the next one, we have global radiation and the stomata opening playing a big role on quite a good amount of days. For drivers on ecosystem respiration, daily ecosystem respiration, soil temperature, the first split, below that. So see how ecosystem respiration goes increasing, but we have some drops like here, here, and there that we're going to explore. This first one below 22.5, so ecosystem respiration go increasing in a combination of soil temperature, water content, the green index, the amount of biomass present in the system start playing a role in there. And over here, I want you to look at this. We had this high number here, and it dropped. Even that soil temperature was higher here, and we have very nice here green index as the main factor explaining this side of the split. And whenever soil water content goes below the wilting point, soil ecosystem respiration drops. And over here, whenever we have soil water content on the other extreme, quite high, it also drops, and something similar happened over here. So far we have annual carbon balance affected by fire, we have seasonal, we have monthly, and we have daily factors affecting ecosystem carbon fluxes. So with that, we can come up with conclusion here that Florida rangelands are in a strong carbon sink, close to 10 megagram of carbon under the 4 years of fire cycle. It's a carbon source during the 5 year. It's a carbon sink even under extended drought conditions. And photosynthetic capacity is recovered with the next 3 or 4 months after fire. and above ground in carbon vegetation is recovered by 4 months after fire. Research future, how fire, grazing, and climatic fluctuations interact. I know that sites are going to be start grazed soon, so that's something interesting to look at. And like I mentioned before, we're working on the warming equivalent based on methane and N2O. And it takes a village. There's a lot of people behind these that I haven't even met yet, and I hope to do that soon. And I thank all of them for their help. And thank you so much. And this is me if you want to contact me later.
Okay, thank you, Dr. Bracho. Now is the time, everybody. If you have a question, please enter it in the questions box, and we'll go through those now. And so your first question is asking about grazing in the area. If you could mention that, is there any grazing happening in the area?
Grazing is happening, yes, of course, and but not where the tower is at now so far in the last 4.5, 5 years. So grazing was excluded from there, but the information that I have is that it's going to start happening soon, and that is going to be really interesting how grazing is going to be affecting the carbon fluxes. Of course, folks down there will have a clear, they have a plan of what's the stocking rate and how is that going to happen and for how long. So that data is going to be registered, and we will analyze carbon fluxes later based on that.
Okay. All right. The next question is from Marta, and she asks if you would please expand upon what are the greatest advantages of being a part of Ameriflux, and in particular about data sharing.
Yes. In the that slide that I show about Ameriflux, one of those links show the flyer with all the benefits that include data storage, which they can store data even in the high frequency resolution, the 30-minute resolution, and all information that is generated related to the tower, so that it can be in safe storage. That's one thing. Two, as mentioned before during the presentation, data is being downloaded quite a lot. So, and out of that, there will come synthesis papers where people from related to this ecosystem, this site, will be part of those. 2, 3, if there is any problem with any sensor being part of Ameriflux, you can request a loaner while your sensor is fixed or recalibrated, whatever. And that is, I had that experience before. They answered really fast. In terms of 2-3 days, you have the sensor at the site, and you can install it and keep collecting data so that you don't lose it. You can request a site visit, and the Ameriflux team comes to your site and stay with you for 2 weeks, and they bring a Robin system, which is a standard system, and they measure simultaneously with your system and make recommendations on what things you should correct, like this sensor needs to be recalibrated, this process that you are using here, you should use this so that we can have better information. They check calibration on your sensors. That is the site visit. They provide technical assistance if you have any problem. You can call them and they can stay with you on the phone as long as you need for them.
Ross asks, what is the tower's footprint size?
Yes, the sensors are at 2.8 meters, and the footprint, I estimated that it's about 80 meters away from the tower. They represent about 80% of the fluxes being measured by the tower. So it's less than 100 meters away from the tower. It represents about 80% of the fluxes that we're measuring. So we have a good footprint at Ross, and we have a good fetch because the unit it's much larger than that. So
okay. Your next question is, why is the first 4 months of NEP in each year the highest? Because of low NEE, or why?
Remember when I showed the monthly ecosystem respiration the first 4 months? So that's not the time of the highest GPP or the highest ecosystem respiration. It's the relationship of both. So the ratio ecosystem respiration to GPP, remember that's what NEP is, the balance of those two. So, and ecosystem respiration is lower, the proportion of that to GPP is lower during those 4 months. So it's not the highest GPP, it's the highest net ecosystem productivity, it's the net, due to a low respiration on those 4 months.
Okay, great. And one more question, and this is from Carolina, and she asks, do you think the ash brings benefits that promote faster regrowth of biomass and faster recovery of the balance in the ecosystem?
The amount of, yes, the amount of nitrogen that was it was kind of a fertilization event, not as when you apply fertilizer, but what we have the data. There were like between 11 and 13 kilograms of nitrogen per hectare after the fire in ashes and semi-burned material, that for sure that helped to the ecosystem recovery after the fire.
Okay, great. That's the last question. And if by chance anyone does have any other questions, please reach out to Dr. Bracho. And do you have any closing remarks, Doctor, before I talk about some upcoming events?
I just want to thank all people that is working down there. They're doing an excellent job, and the help is really, really great. And I just keep, working in collaboration with everyone down there. And I think this is going to be part of a nice study, and synthesis papers are going to come up after this. And we have to just keep going on this. Thank you.
Wonderful. Well, thank you so much for being our guest today.
Thank you.
All right, everybody. I'm going to share with you about some upcoming events, and then I'll let you go. For one, our upcoming ONA highlight for November is going to be a graduate student ONA highlight with Liz Palmer. She is a PhD student in the animal science program, being advised by Dr. Philippe Moriel. She will be presenting pre-calving nutrition of beef females. And then in December, we'll have another student highlight. Wes Anderson, who is a PhD student in the Wildlife, Ecology, and Conservation Department, will be presenting impacts of an invasive ecosystem engineer upon wetlands and aquatic communities across a subtropical agroecosystem. And Wes has been advised by Dr. Raoul Boughton, and he will be graduating in December. Here is the next upcoming livestock genetics and genomics webinar series. These have been ongoing since September 30th, and it looks like there's going to be one tomorrow, genetics of hair coat shedding in cattle. And then October 21st, fescue tolerance, role of genetics. And October 28th, genetics of feed efficiency in beef cattle. And November 4th, using genome editing for livestock health. And so those are the ones coming up between now and our next webinar, but we'll share about this again next month. Later on this month, we are going to be having our 3rd annual Nutrition for Beef Females event, which is put on by Dr. Philippe Moriel and Dr. Mario Benelli. Philippe will be presenting Supplementing Beef Females, Benefits to Cow and Calf. Dr. Benelli will present Boosting the Reproductive Efficiency of Beef Females. This event is going to be online through Zoom on October 22nd at 6 p.m. There is no registration required. You just need to remember this link shown here so that you can connect to the event that evening. If you have any questions, please reach out to Dr. Moriel. I know it's a ways off, but just wanted to make sure you knew about the Florida Cattlemen's Institute and Allied Trade Show. This year, the event is going to be online, a virtual program, and it's going to start at 1 p.m. and go through the afternoon. And then also kind of out there in the near future, April 1st, we're going to be having our next field day, and it will also be a celebration of our 80th anniversary. And we do hope to be in person, but time will tell. So look for details on that coming out around the 1st of the year. And so I believe that's all I have. If you would like to have the registration links for those events or more details on them, please visit our website shown here on this slide. And if you're not already doing so, we invite you to follow us on Facebook or Twitter. Also check out our YouTube channel where we have many, many awesome videos for you to see. And if you do not receive our weekly news blast by email, use this email shown here to send me a message and let me know that you'd like to be added. So thank you so much again for joining us today. And Dr. Bracho, thank you so much for being with us as a guest.
Thank you.